we should have thought about it. Usually we try to take as many factors as we can.
It's kind of important because, for example, it is in Austria where I am, but it's not in Germany. Germany was yesterday. So I think it's fully impossible. I mean, there's so many different possibilities. So I think like it's not going to work.
Yeah, like Jen and I... Sorry, go ahead.
No, what's the holiday today for some of you?
It's the All Saints. So it's a Catholic holiday. So I mean, I think they kind of, it's like, I wouldn't say it's a Halloween for Catholic people because Catholic people could be pissed if say something like that, but you mostly celebrate like this concept of Passover, I mean, death and birth, I don't know, is there. So today is the All Saints in the Catholic country.
Okay, got it. Well, as always, we will have these up on YouTube as long as that's okay with both of you. And so people always catch up later. Lots of people do that. All right, so we're set on YouTube. We got the email sent out. So whenever you want to...
I think we should start because it's 11:02 and we have to
Yes, share my...
Yeah, while you do that, I'll introduce...
Okay, I hope it's visible.
Yep.
Fantastic.
Perfect. Okay. Welcome everybody for another session of Cell Migration Seminar. Today we have two wonderful speakers, Stefania and Emma. We will first go ahead with Stefania, who did her undergrad in University in Milan, Italy. Afterwards, she joined the lab of Weiland Huttner at Max Planck Institute and Genetics in Dresden, where she studied the regulation of cell delamination and its link to transition from apical progenitors to basal progenitors for her PhD in the developing mammalian neocortex. And currently she is a postdoc fellow in Carl Philipp Heisenberg's lab at Institute of Science and Technology, Austria, where she is working on how substrate modulates lateral mesendoderm migration in early zebrafish gastrulation. So I will give this floor to Stefania and really thank you for accepting our invitation and presenting today your work.
Thank you. Thanks for the kind introduction and for giving me the chance to talk about my work. So today I will talk about two novel roles of the ectoderm in the early zebrafish gastrula. I'm sorry if my voice is funky, but like I got sick after a conference, so I didn't fully recover. So I hope it's going to be clear enough anyway. So let's get going. Okay. We all know that gastrulation is important. I think that's a matter of fact. And here you see gastrulation, the schematic in three different organisms. They look really different among them, but they all have a common denominator. There is amazing change in shape that are driven by tightly controlled cell movement and intercalation. In Heisenberg lab, basically Heisenberg lab, we use as one remote system zebrafish, and the zebrafish gastrulation starts when the p-body reaches the 50%. At this point, the cells that are at the margin will ingress and migrate between ectoderm and yolk cell. Now, in this video, you can see mesendoderm in green. You will see here you have the dorsa, and most of my talk, the dorsa will be actually on the right side, and the animal pole on the top. You will see the cell moving towards the animal pole to then converge towards the foreign body axis. Now, for my work, I'm interested in the lateral mesendoderm, that is the one you see here towards you in the screen. And the reason why I'm interested is because it has a fairly complex migratory behavior. In fact, after ingression, initially moves towards the animal pole in a single cell collective manner. However, it never reaches the animal pole because amid gastrulation, it starts to lose speed and also start to have a kind of wobbling motion that then precedes the convergence. Thanks to the work of many scientists, we know many factors that regulate convergence, but we know little about the first part of the migration, especially where the cell never reached the animal pole. What stops them from getting there? And this is my question. As I was saying in the introduction, I'm trying to assess it, to address it by having a look to the substrate, asking whether substrate modulates lateral mesenteric migration. This is the autonomy talk. The first part will talk about which is the substrate, or what these cells use, and that where they actually modify the migratory behavior. Now, one interesting thing about early zebrafish gastrula is that ECM doesn't seem to have a role. In fact, at this stage, so before onset of convergence, there are only few puncta visible that has been shown already a while ago. The people already shown that the lack of fibronectin and integrins affects morphogenesis only later, pointing to the fact that the cells don't use ECM for migration, so they have to have a nice substrate. And now we have two possibilities. They can either use the membrane of the ectoderm or the membrane of the yolk, but they are like in a sandwich between the two. To assess which is the main substrate, I use different means. I had a look to what the cells will touch more often, to what the cells will adhere, and then if I force them to choose between one of the two, which one they would prefer. Now, going to the directionality of the protrusion, the way I had a look at them, I did like sparse labeling of actin that you can see here in white. And then I had a look at the cells that you will now see coming here, migrated towards the animal pole. I had then quantified, you can see this is beautiful migrating cells that are mesendoderm. And then I had a look to the protrusion, actin protrusion, I had a look to whether they were towards the ectoderm or towards the yolk. As you can see, this quantification, regardless of the protrusion length from the cell mass, the majority of the protrusion are directed towards the ectoderm. Say that at least the contact more the ectoderm compared to the yolk membrane. Now, obviously, protrusion directionality is not exactly adhesion nor migration, so I wanted to see what the cell adheres on. And so to do that, I actually had kind of a quick, to me, nice experiment. So I took kind of a snapshot when they were almost through the migration, and they peeled away the yolk membrane and the blastoderm and fixed them immediately, and then asked, okay, where do I find the cells? Where do I find these lateral mesendoderm cells? As you can see here in this example, the mesendoderm is always in green. You can see the majority of the cells are not on the yolk membrane, that you can see these are also yolk sensation nuclei, but they're actually at the blastoderm, suggesting that they are adhering to the ectoderm. But this is a fixed endpoint. I wanted to see it live because that's what was interesting me. But the problem, as I say, that they are in a tight sandwich, so it's kind of impossible to discriminate. So the way how I went around it was actually to create space. How you create space, you can inject mannitol. Mannitol is in sugar, but increases the interstitial fluid. And when you have more interstitial fluid, this will be between the ectoderm and the yolk. And now you can see the cells cannot be on both sides. They have to be either one and the other one. And the other amazing thing is they don't care. They do migrate as there would have been no mannitol, so the migratory behavior doesn't change at all. So now by live, I can score throughout this animal migration whether I found them more towards the ectoderm or towards the yolk. And this is a later time point of the same video. I think you can really appreciate here down that most of the cells are on the ectoderm. And this you can see in this quantification why throughout this time of migration, which is roughly an hour and a half, a bit more, the 70% of the time, on average, the cells are on the ectoderm. Altogether with the protrusion, the addition assay suggesting that the ectoderm is the main substrate. Okay, that's interesting. Actually quite new because we didn't know. It's also quite funky because actually the ectoderm moves in the opposite direction of the migration. But is there any difference? So especially when we have a look to the cell that reach the animal, the move towards the animal pole, is there any difference whether they use the ectoderm or the yolk membrane? And so
I need something clarified. Does the yolk have any membrane over it, or what is enclosing the yolk?
It's a membrane. It's
like a plasma membrane?
Yeah,
okay.
It's a plasma membrane.
Thank you.
Yeah, sorry. So what you see here is actually injected dextran dye in the yolk after the yolk. So how it forms, sorry, I haven't clarified that. Now how the white cell, that is a yolk interstitial layer, is formed, that blastoderm cells like drop on the yolk and they can fuse on it. So the membrane that you see here, it was mostly a membrane of these cells before they fuse with the yolk. And there are nuclei here that are, they were nuclei of the cells, and then from syncytium, we have also cortex. And for example, we know that the epibole is driven by the actin flow at the cortex, that they are the external wire cells. So we have a membrane there. Okay. So going back, so I wanted to see if actually there was any difference. So I had a look at the cells that were either the most or the least on the ectoderm, and see if there was any difference how the cell migrated. And first of all, I had a look to that one of the characteristic features of migration, which is persistent. And interesting enough, with the cells that are on the ectoderm, whether we had a look to the lateral mesendoderm as a population there, or at the endoderm alone, they are more persistent when they're on the ectoderm. Then we had a look at the directionality, especially because I wanted to know if what the cells used to reach this most animal part of the embryo. And interesting enough, both for lateral mesendoderm or endoderm alone, the directionality is strictly towards the animal pole if they use ectoderm, while this is kind of lost when they use most of the yolk. And this you can see by the fact that displacement towards the animal pole is the highest for the cells that are on the ectoderm compared to the one that are mostly on the yolk, suggesting that the animal-directed lateral mesendoderm cells use ectoderm as a substrate. So going back to the slide, to this introductory slide, now we have an answer to the first question, is the ectoderm. And now we can ask whether the ectoderm modulates lateral mesendoderm migration. Now, if you think about the fact that the cell migrates towards the animal pole but never reaches it, one possibility that the ectoderm there at the animal pole is different from the one on the lateral side. So like there is an animal vegetal patterning. To test this hypothesis, I used transplantation, which is something that we can do easily with zebrafish. So I took ectoderm from the animal pole or from the lateral side, and then transplanted the host embryo on the lateral side, then had a look how the cell would migrate when they were encountered the transplanted ectoderm. And here the example, and here you can see in blue the lateral ectoderm transplants. I hope we can appreciate that the cells, maybe they feel that there's some cells more, but they kind of find the migration. But now if you have a look to the animal ectoderm transplants, I want you to have a look here, you can kind of see this void that initially is there. Actually, the cell can kind of move, like they kind of cover up from the side. And that we can quantify this kind of inability to move towards the anterior pole. In fact, now if you see the lateral to lateral ectoderm transplant, is pretty much the displacement towards the anterior pole is equal to an untransplanted embryo, but this is strongly decreased when you put an animal ectoderm on the lateral side. This we can also see as percentage of cells that reach the most animal part of the embryo, which decreases from a normal roughly 70% to down to 20%, a bit more than 20%. Altogether suggesting that animal ectoderm inhibits the lateral mesendoderm migration towards the animal pole. Okay, that's perfect, but what's the difference? And when we think about substrate migration, we know that how the substrate is, for example, from the point of view of stiffness, softness, can really affect the ability of cells to migrate on it. So I kind of wanted to have a look if the properties of the substrate, so the properties would be different along the animal vegetal axis. And the first thing I had a look at was actin cell cortex. And here you can see the nuclei in magenta, the actin cortex here. And what I want you to focus on would be the formation of an interface between the migrating lateral mesendoderm and the ectoderm over time. And you can kind of see it here. You can clearly see that there is an accumulation of actin between the lateral mesendoderm and the ectoderm. What is even more interesting that if you follow from the margin towards the anterior pole, this accumulation decreases over time. So more you go further, less actin is there. And this you can see in these plots in which I quantify the actin intensity. Within the tissue, there is no difference actually along the animal vegetal axis, but really at this where the cells are migrating on. So this interface, you have higher accumulation close to the margin that they drops even below the amount of actin you will have within the cells. And we know that actin accumulation at the cortex can modify how, for example, the membrane properties of the however, suggested that could have a difference, at least in the properties of the membrane of the cortex closer to the margin. Now, I want also to have a look more at the tissue level. Have been shown several reports recently that the rigidity of the tissue or the fluidity tissue really depends on how the tissue is compact, how many contacts the cells form between each other. And here you can see the animal ectoderm, the lateral ectoderm, two embryos that have been imaged at the same time. Here you can see the lateral mesendoderm that will start migrating towards the annual pole. And I hope you can appreciate that over time, the lateral ectoderm gets more and more compact, while the lateral ectoderm stays loose with all this interstitial fluid between cells. And this you can quantify it as a fraction, where we have a really high fraction in the lateral ectoderm, and we know from other studies that the cell fraction correlates with tissue rigidity, suggesting that most probably the tissue at the lateral side, at the ectoderm at the lateral side, might be more rigid, like compared to the animal ectoderm. But if that holds true, we could think about that too. We could change the effect of the ectoderm on the migration just by changing the property of the cells. For example, we could turn animal ectoderm in lateral ectoderm by increasing cell contractility using considered active array, so making the cells more stiffer, or we could change lateral ectoderm by decreasing contractility using considered active myosin phosphatase. And that should be enough to change the effect on the migration. And that's what I did. So in this case, you can see here in red, I took animal ectoderm from a host in which I express a small amount of considered active array. And now I hope you can appreciate that the cells migrate happily, like there was fully normal type of ectoderm. Here in the light blue, we can see the opposite. We can see lateral ectoderm, which I decrease contractility with the considered active myosin phosphatase. And similar to the normal animal ectoderm, we can see now that the cells don't really like to use it as substrate for migration. As before, I showed you before, we can quantify this effect. And now, for example, you can see we fully recover the displacement towards the annual pole on a more contractile animal ectoderm, and we decrease it on a less contractile lateral ectoderm. And also, when you have a look to the cell that reach the most annual part of the embryo, we fully inverted. The animal ectoderm goes from 20% to almost 70%, the lateral ectoderm to 70% to 20%. Pointing that is enough to change one single property of the cells to fully change the ability of the lateral mesendoderm to migrate on it. That, which is extremely cool because it's actually, I wouldn't expect it was so easy. Now, but what makes a difference between animal ectoderm and lateral ectoderm? What sets the difference? And to try to answer this question, I wanted to have a look actually if I could see a difference at the transcriptome level. And for doing so, I did back-seq of the animal ectoderm or the lateral ectoderm, and we can see genes that we know are expressed on the animal ectoderm, like OTX2B and A or WNT11, WNT5B. Among the genes expressed at the lateral ectoderm, there is cording, which is a BMP inhibitor. And then we had a look back to how the morphogen are in the embryo, even before the onset of gastrulation. And this is a small schematic taken from review, and you can kind of see that, for example, BMP is higher on the ventral side, they're decreasing towards the dorsal side. But if you have a look at the gradient, at the morphogen from the animal to the vegetal pole, we can see that closer to the margin, even before onset of gastrulation, we have several morphogens. We have nodal, we have GF, we have WNT. But at the animal pole, the main morphogen presence is BMP. And we wonder if the presence of mostly only BMP there could actually set a difference at the animal pole. And we also know that BMP is important for gastrulation movements in general. So to test that, we first remove BMP in the full embryo. Here you can see a control. You can see how the cell, now we see the animal pole, sorry, I didn't mention it initially. So this is the animal pole view. And you can see the cells never reach the animal pole before convergence. Now, if you have the same view of the BMP depleted embryo, you can clearly see that the cells don't stop migrating. So whatever was there stopping them from migration isn't there anymore. And they actually cells, you have cells that will form the trunk, now colliding to the future head. But obviously this is a depletion in the full embryo, so you also affect mesendoderm per se. So to test whether the effect was specific to the ectoderm or depended on the mesendoderm, we went back to the transplantation setup I showed you before. So now we have again animal ectoderm, but in this case, it's animal ectoderm which I depleted one of the main BMP ligands, BMP2B, and then I took this animal ectoderm and transplanted in a wild-type BMP expressing host. And I hope you can now appreciate that the cells actually, again, migrate pretty much happily on the animal ectoderm. And this, as I showed you with the quantification I showed you before, we can see that we fully recover the ability to displace towards the animal pole. And also we partially recover the ability to reach the most animal part of the embryo, suggesting that in the BMP deficient animal ectoderm is now a permissive substrate. And with that, I want to conclude my talk with a summary of what I showed you. I showed you that ectoderm is a substrate, especially it's a substrate that is used by the cells to go towards the animal pole. There is some difference in the ectoderm because the mesendoderm decrease the ability to move to animal pole when it encounters the animal ectoderm. And we think the difference is within the tissue property, where we think that we can see that the animal ectoderm have different tissue properties. And moreover, we can also see my slide freeze. Can someone say if my presentation is still ongoing?
It is going, yeah.
Okay, so my slides just freeze. Okay, no, sorry. So it's sufficient to just change the contractility of the cells to invert fully the effect of the ectoderm. And finally, a BMP signal is important to define the non-permissiveness of the ectoderm at the animal pole. So now what we are trying to really put together is the, let's say, the part of the tissue properties, the BMP, to see if actually BMP is controlling the difference in the tissue that we see between animal and lateral ectoderm. And with that, I want to thank CPs, and first of all of you guys for being here, and CPs to be my supervisor throughout this project, the people in the lab, especially Shin, Suryas, and Alex, Saren from the 6th lab, because if you're seeing amazing graphs with a lot of tracks into this graph was thanks to a software he created to use R, and it's really super cool. So if you're interested in that, contact him or me. And then the imaging facility, the fish facility, scientific computing facility, and NGS facility, and I'm happy to take your questions. I think I stop also with the sharing.
Thank you so much, Stefania. We can first go with Claire. She has her hand raised.
Hi there. Very beautiful sort of old school talk.
Thanks.
So are you worried about what, I mean, this is cell-cell adhesion that's driving this, don't you think?
I mean, this is one possibility. At the moment, we don't know, honestly speaking. I haven't focused on what it is.
Well, I thought you said there was no ECM.
There is no ECM. I cannot exclude that is not cell adhesion. I cannot exclude that there is some membrane friction. The point I cannot exclude.
Yeah. Okay, so no zero, like no non-receptor mediated, right?
Yes.
I mean, are there cadherins in there if you stain them?
There are cadherins there. That's definitely for sure. And I have, so if there is contacts for me, they're really, really small and transient because I haven't been able to see it on EM or not really careful search. So normally you kind of see the contacts by EM if it's ester contacts, but if they're there, there is more transit, so that I really have to scan many sections of the cells to really find them.
They're under a lot of compression in there, right? So you think friction would be sufficient?
I mean, the point that actually they migrate also without compression, because when they increase the space, there's no compression anymore.
Right.
So, but doesn't mean that there is no some sort of transit addition that allows a slippage of the membrane. I mean, the point that we don't really know at the moment. There is a few experiments that has to be done. It's a bit hard to manipulate Ectoderm without screwing up the full embryo, because obviously what it keeps them together.
Yeah.
But there are a few experiments I hope to do. I mean, I hope a certain point that can be done to kind of really pinpoint what it is. But I'm not sure if that was your question.
Yes.
Because okay.
Okay, great. Thanks, Claire. Thanks, Day. Dylan, do you want to go next?
Yeah, thanks. So thank you for the talk. I just have a quick question about the last part, which is the BMP. I was quite confused about the timing, because in Xenopus, we have the BMP antagonists are secreted like cordon and noggin prior to the migration. So I don't know about zebrafish, how that happens. So I think that by the time that is the same timing. So
I mean, they are present earlier. They're definitely present earlier. BMP gradient, cordon expression. I mean, cordon is one of the dorsal. It's like high expressed at the dorsal, and it's important to define the lack of BMP at the dorsal side. And we do see still express. Actually, was semi-surprised to see at the lateral side. I guess the reason why I have never seen much at the lateral side, that the dorsal side is so strong that when we do in situ, most likely we at a certain point we stop much earlier. But all stuff is set up earlier. So the BMP, the BMP grader is not something that comes with the migration. It's there much earlier. It's important to set it up. Also the mesendoderm fade ventral to dorsal. So it's not something, it's already there. So the idea would be that the fact that it's there, especially is there, let's say, and the annual pole, for what is my knowledge, is no other main morphogens there. Maybe makes the, I mean, we know from other work in convergence, for example, that BMP downregulates Ectoderm. That has been shown during convergence. And so one possibility, for example, that the fact that you have only BMP there, that is enough to kind of make the cell less adhesive to each other, so looser to, so more for that reason. Kind of the tissue process after. But we still, there's a few stuff have to be tested to be fully sure about that.
Yeah, I just, so the question is like precisely in the timing manner, like is the migration starts with the secretion of these antagonists or after?
No, it was that it's already there. The antagonist is already, I mean, for sure,
they're already there. I see.
I mean, the Tuscalone has been done later, because the Tuscalone obviously I did at the time of the migration, but at the dorsal side is fully there. Whether it's there at the lateral side earlier than that, I don't know. I can tell you that for sure I see it around 60-65%. That is when they migrate towards animal pole. But the dorsal side, the expression is already there. I mean, it has to be there.
Yeah. All right. Thank you.
Sure.
Awesome. Thank you, Dylan, for your question. Our next question is from Gaurav Bharadwaj, asking, it's like a clarification question, which cell population contributes to actin cortex? Is it from ectoderm or LME or both?
So I would say it's ectoderm, for the simple fact that the lateral mesendoderm cells express so little anything you inject that I think from the video you could see that almost you couldn't see them when they were migrating. So my guess is that what we see as accumulation is ectoderm, simply because for reason I not known to myself, once the lateral mesendoderm cells ingress, they kind of deregulate any mRNA or any maternal mRNA. So you can inject nuclei whatsoever, and they drop as soon as it ingress, which makes it quite funky when you want to image them. But yeah, so I would say just because of expression amounts, at least that is most likely ectoderm.
I actually had a quick question, and this is again a clarification question. In your animal versus ectoderm, and I think it's Nicolata's work, the rigidity differences you see, is it due to like division of the cells? Like how is it controlled during development?
No, the point is that the animal ectoderm, it kind of stays that loose. It's already like that. Why it's like that earlier, I don't know. I don't see difference in division. It just kind of, the amount of interstitial fluid you see is already like that. Why the
so you think it's cell adhesion actually, that just cells have those spaces?
Could be cell adhesion. I mean, that's what we know that can influence that. Could be something else which I don't know at the moment. But the lateral ectoderm actually gets more and more compact. So actually, the cell fracture gets to 97%, which is almost no fluid around the cells. How that get, I don't super sure. I will guess maybe to the fact that I don't know that there's accumulation cells over there, but I don't think it's cell division because I don't really see like more cell division, less cell division. At least nothing that is like pop-ups, like, you know, that you say, okay, this is clearly dividing more than the other one.
Awesome. Thank you so much.
All right, great. Thank you, Stefania. That was fantastic. If anyone has further questions, feel free to drop them in the chat or follow up after our next talk. Emma, whenever you're ready, go ahead and share your screen. So I'm really delighted to introduce our second speaker for today. We have Dr. Emma Lång. She received her master's in molecular biology at the University of Umeå in Sweden and went on to do her PhD in Professor Tone Tunjum's research group at the University of Oslo. Following the completion of her PhD, Emma joined the group of Dr. Stig Ove Bo at Oslo University Hospital. And her recent interests, she's done a lot of different things, but her recent interests are in understanding emergent dynamic behaviors of cells during wound repair development and cancer progression. And so we'll hear today about her work that was, I believe, recently published in PNAS on collective migration and quiescence. So those of you who know anything about me know that I'm extra excited for this talk. So Emma, the floor is yours. Thanks for being here.
Thank you, Jennifer, for a very nice introduction. And thank you both for giving me the opportunity to present our work here today. So I am interested in mechanical and biochemical processes that regulate cell migration during tissue development, wound healing, and cancer metastasis. And today I will present our recent work on keratinocyte monolayers and the dynamic cell behavior that is activated during transition between a quiescent and a proliferative cell state. So cells in the human body or in any other organism that has the ability to divide can be either in a dormant, non-dividing cell state, referred to as quiescence, or the cells can re-enter the cell cycle and divide again. And what I would like to emphasize in this presentation is that cells that enter quiescence have a tendency to become more static and less motile, while cells that re-enter the cell cycle often display an increased cell dynamic behavior. And there are several examples of this in nature. So increased dynamic cell behavior is observed during wound healing as basal keratinocytes enter a proliferative state at the same time as they begin to migrate into the open wound bed. And in addition, cancer cells can enter a dormant quiescent-like cell state and stay there for several years. And when these cells eventually decide to exit quiescence, they often become highly migratory and invasive. Furthermore, immune cells in the lymph nodes are reactivated and begin to migrate as a response to mitogens that are produced during infections in the body. And finally, increased dynamic cell behavior is also associated with reactivation of quiescent stem cells during tissue regeneration and repair. So we have developed an experimental approach to study quiescent-dependent cell motility in vitro. And in our experiments, we use a human keratinocyte cell line called HACAT. And these cells express M-Cherry-tagged histone H2B in the nucleus. And we use this nuclear signal to track the cell movements in our experiment. So first, we seed human keratinocytes into confluent monolayers in 96 well plates. And each well is approximately 7 millimeters in diameter. So the cells are confined to this area. And the movie here show our experimental system. So as I just told you, first we seed human keratinocytes into confluent cell sheets. Then we reprogram all the cells into quiescence by serum starvation. And after a certain time period in quiescence, we re-stimulate the cells with serum-containing media. And as the cells exit quiescence, we observe a highly coordinated collective cell migration response that we can monitor by live cell imaging. And this movie and the graph here to your right show the level of cell sheet activity in the 3 different stages of our experimental system. And what I would like to emphasize here is that this collective cell migration is only activated if the cells have spent a certain time period in quiescence before serum restimulation. So this dynamic behavior is only observed as cells exit quiescence. So the dynamic behavior that we observe after serum restimulation of quiescent cell sheets is remarkably consistent. So we observe a flow of the cells directed inwards towards the central part of the monolayer. And this leads to an increase in the cell density and the cell sheet thickness in the central part of the monolayer, while it leads to a decreased cell density and cell sheet thickness closer to the edges. Consequently, cells in the central part of the monolayer become more compressed, while the cells closer to the edges become more stretched out. So this directed cell migration leads to a redistribution of cell mass within the confluent monolayer. So the question we want to answer is, how is this dynamic behavior activated during exit from quiescence? So to answer this, we perform traction force microscopy experiments. So here we have extracted both traction forces and intercellular tension from a microscopic field before and after serum restimulation of a quiescent monolayer. And we observe an instant increase in both the traction forces and the intercellular tension as cells exit quiescence. And if we look at the time evolution of these forces, illustrated here by these chymographs, we see an instant amplification of forces after serum restimulation of the quiescent cell sheets. We also see how the force magnitudes decrease at the same time as the cell sheet velocities increase. And the same data is plotted here in these graphs. So where we've plotted traction magnitude and tension in red and cell migration speed in blue. So again, serum re-stimulation leads to an amplification of forces. These forces gradually decrease as the cells begin to migrate. So in our system, we observe an anti-correlation between forces and motility. So if we then compare the force maps before and after serum re-stimulation, we notice that the amplified forces produced as cells exit quiescence originates from forces already present in the quiescent cell sheet. And this is also observed in these chymographs here. So again, there are forces in the quiescent cell sheet, and it is these forces that become amplified after serum re-stimulation. So we think that this in part explains how a monolayer is able to mount such a rapid and coordinated amplification of forces as cells exit quiescence. And this means that the mechanical system that generates these forces is already present in the quiescent cell sheet. So now we wanted to address the role of actinomyosin in generation of forces and motility after exit from quiescence. And so to answer this, we treated quiescent monolayers with inhibitors that block the function of actin or myosin. And here we see how these inhibitors are able to prevent the force amplification after serum re-stimulation. So shown here by the red line. We also see how these inhibitors are able to prevent activation of cell migration. So the results show that actinomyosin activity is required for the initial amplification of forces as cells exit quiescence. And the results also suggest that this force amplification is linked to activation of cell migration. So to follow up on this, we wanted to examine how inhibition of cell migration affects the amplification of forces that we observe. So here we have treated quiescent monolayers with inhibitors that are known to inhibit cell migration. So we've used an inhibitor against RAC1, we've inhibited the EGF receptor EGFR, and ERK. We've also included 2 inhibitors that are specific for focal adhesion kinase. So if we first look at the lower panel where we plotted the cell migration speed, we see how all these inhibitors prevent activation of cell migration. If we then look at the traction magnitude or the forces produced after serum re-stimulation, we don't really see an effect on the initial amplification of forces. However, we do see that these inhibitors are able to reduce the force decay rate in the monolayer after serum re-stimulation. So these results suggest that the force decay is coupled to activation of cell migration. So now, in order to understand how stress distribution in the monolayer can lead to activation of cell migration, we developed a minimal mathematical model based on active gel theory. And so this model includes essential physical components in the monolayer, such as cell deformation, cell substrate friction, and actinomyosin-mediated stress. So we performed numerical simulations where we tried to mimic the experimental conditions. So we used a circular domain with a random distribution of actinomyosin activity giving the pre-stressed state of the monolayer. And so these three movies show the dynamic behavior produced in the numerical simulations when we used different levels of the initial pre-stress. So here we have an increase in the initial pre-stress from left to right. And in the graphs below here, we've systematically increased the initial pre-stress and examined how this affects the forces and the average radial speed produced in the simulations. We see how an increase in the initial pre-stress leads to both an increase in the traction magnitudes and also the average radial speed produced in the simulations. So what these numerical simulations told us is that a certain level of initial pre-stress is required in order to generate a dynamic behavior that resembles what we observe in our experiments. And so the theory predicts stress amplification as a key component for cell sheet displacement. And now we wanted to test these predictions experimentally. And so to achieve this, we took the advantage of something that we call quiescent depth. And with quiescent depth, we mean the time the cells spend in quiescence before they are woken up and reactivated to enter the cell cycle again. And we assumed that the length of serum starvation would determine the stress produced as cells exit quiescence. And this turned out to be true. And so here we have measured the tension in the monolayers after serum restimulation of quiescent cell sheets that has been exposed to different length of serum starvation. So we see how an increase in the starvation length leads to production of increased tension in the monolayer. And if we look at the cell sheet velocities produced, we also see how an increase in the starvation length leads to a production of increased cell sheet velocities. And the experimental values fit well with the theoretical predictions shown here by the red line. So both the level of stress and the cell sheet velocities produced after serum restimulation correlates with the quiescent depth. And in summary, both our theoretical model and the experiments show that a monolayer has to be able to generate a certain level of global stress in order to activate an efficient cell sheet displacement. So now to the take-home message from this study. Quiescent monolayers are poised for mobilization. Dynamic behavior after exit from quiescence is facilitated by stress amplification. The stress magnitude and dynamic behavior after exit from quiescence is controlled by quiescent depth. So there exists a mechanical system in quiescent cell sheets that are ready to become activated on short notice to produce dynamic cell behavior. And so finally, I would like to acknowledge all the people that have contributed to this work, especially our collaborators at the Department of Mathematics, University of Oslo, Christian Pedersen and Andreas Karlsson, who have developed the mathematical model and performed numerical simulations. Of course, everyone in Stig Ove Bø's research group, our funding partners, and with that, I thank you for your attention, and I am happy to answer questions.
Awesome. Thank you so much. That was fantastic. My lab is going to read your paper for our next lab meeting. I'm really, really excited about so much of it. Ste, why don't you go first?
Yeah, I actually have two questions. The first one is, what will happen if you kind of erase the state of difference by doing lateral cooling treatment right before, wash it out like before serum? Like, can you kind of erase this different stress between cells? If so, they will just recreate the same pattern, or they will create new pattern, how the migration will work? And that will be the first question. And the second question would be if you could actually have a look to this pattern with, for example, Fuji cell cycle, to see if this pattern depends on when they re-enter in S phase. Also, like if there is a correlation between the time it takes to this, like how the stress pattern are, if you can actually, aside the fact that they re-enter in the cycle, if they actually there is difference in the timing between neighboring cells.
You mean a difference in when neighboring cells enter mitosis? Oh, that's really interesting. We have studied how the length of starvation affects the time it takes for like the whole cell sheet to begin to divide again. We haven't really looked at single cells like neighboring cells, if it's different between those, but that could be interesting to see. I'm not sure if I understood the first question.
Yeah, I was wondering if you could kind of erase these pattern stress forces by treating with latrunculin or any other cytoskeletal inhibitor ahead of the serum. So you treat it, you wash it out, and only after you give the serum. Will be these pattern stress forces like erased, so they will be like equal between each other? If so, what would be created? Would be a different pattern, or would be any?
So we did use latrunculin A, and that completely blocked the amplification of both the forces, and it completely inhibited cell migration.
Yeah, I was just wondering if you use it before or remove it before the serum.
So remove it before? Yes. Oh, we haven't done that. No. So that could be interesting to look at. Thank you.
Awesome. There's a question from Ankit, unless you want to ask yourself, Ankit, since I see you on the video here.
Yeah. So very nice talk. So I was wondering, is there any way this quiescent depth can be correlated with the stem cell activation or immune cell activation? Have you tested this?
No, we haven't tested the method on stem cells or immune cells yet, but it would be interesting to look at how these cells are affected by this serum starvation and then reactivation. Mm-hmm. But we think that it's a common that at least it might be a common mechanism that when we induce quiescence in these human keratinocytes, that it would resemble like stem cells in the skin, for example, and how these cells then are reactivated in different situations.
Great, thank you. Jeff, why don't you go next? Nice to see you.
Yeah, good to see you, Emma. Thank you. It's a very nice talk. I like what you said. I have two questions. One is, have you measured any indices of cell shape and cell shape changes during these phenomena? And I'm just wondering also if you saw any evidence of wave-like behavior that's been reported before. But really the shape changes I'd be curious about.
Yes. Oh, thank you. I'm happy that you like to talk. So yes, we have looked at the cell shape changes. We try to both study the form, like if the cells become elongated or in any way change shape as they begin to migrate. And we also looked at this perimeter the cell perimeter, and tried to connect it to this unjamming-jamming transition. But we really haven't found any significant data on this. No, we can't really say that it's any shape change or a change of the cell area that activates these migrations. We think it's something else. Yeah.
Okay, thank you.
Thank you.
Awesome. Thank you, Jeff. Anna, do you want to go next? And then there's a question from Nile we will take.
Yes. Oh, very nice talk. I have a question. So we know that when we do extraversion of cells, we disassemble adhesions and stress fibers. So when you add back the serum, did you see a correlation between formation of focal adhesion of stress fibers and forces?
We have looked at actin and also stained for focal adhesions using Paxilin. So we first, to answer about the focal adhesions, we do see an increase in size when we look at Paxilin staining. So there is definitely something, some changes in the focal adhesions after serum restimulation. And we also see a lot of actin reorganization in the cell sheets, both on the basal side and on the apical side of the monolayer. So it's definitely, I think it's these remodelings that probably generate this amplification of forces that we observe in the traction force microscopy experiments.
Thank you.
Great. We have two questions from Nile. First asking so these are technical questions, how were the tensions calculated from the traction maps?
That I can't really answer right now, I think. We used, I can't really say that now. I'm sorry. I need to go back and look at the methods. Yes, I must admit I didn't do it myself. So yeah, but I can answer that on the email or yeah.
Yeah. You may have used monolayer stress microscopy, which was developed by Jeff Redberg's group.
Yes, we did. But how, I can't really tell right now.
And Nile is also asking how the quiescence depth is calculated.
So that's, we use the different length of starvation, and that's what we call quiescent depth. So we've actually done experiments where we starve the monolayers for different time periods before serum restimulation, and then we detect the different dynamic behaviors. So we actually know that there's like a border, like after 2-3 days of serum starvation, we are able to activate this dynamic cell behavior. But if the cells are starved for a shorter time period, we don't. So that's what we mean with the quiescent depth, like the time of starvation.
I think your quiescence depth may explain some things that I've been confused about in my own system, and things that haven't worked the way I thought, and I wonder if it has to do with this. So this has given me a lot of things to think about. I was wondering, okay, I have like 1,000 questions, so other people should pop in. Otherwise I might just start. But I'm just wondering, in terms of sort of measuring quiescence, we always talk about it just as like, it's a lack of proliferation, but are there markers? Are there ways that we could say these cells are like more quiescent than those cells, or is it really, do you think about it that same way of just, okay, the cells aren't proliferating? How do you think about this?
And no, I agree with you. That's how we at the moment can measure it. So we see that the cells stop dividing. And then when we re-stimulate them, we see that they begin to migrate, and then eventually they also begin to divide. I think it's been discussing in quite a lot of papers what is a true marker for a dormant cell and a dividing cell. So yeah, it's definitely something we should find out. Maybe it's a way to look at like mRNAs expressed in the cells that can show if a cell is like more dormant than the other, like look at the transcriptomic profile.
Yeah, it would be nice.
Interesting.
It'd be convenient if there was a single marker.
Yeah.
Denor, go ahead. Looks like, okay, great.
Hi, Emma. Very nice talk. So I have
Thank you.
2 questions, curiosity questions. So when you say the cells have undergone deeper dormancy, so how long does this deep dormant cell take to reactivate themselves to dormancy and then begin to migrate?
So the time it takes for them to begin to migrate after serum re-stimulation?
No, first bringing back this transcriptome to its normalcy, that is being coming out of quiescence, how long does it take? And after coming out of quiescence, that is when it becomes like a normal cell, then it starts to migrate, right? So I am interested in the period where it comes back to normalcy. So what is this time delay that it takes for the cells to exit quiescent when they are undergoing such a deep dormant state? So that is my question here. So before migration.
Yeah, it's an interesting question. You know, we've only looked at the migratory process and of course the traction forces as the cells begin to migrate. So I can only tell you that between 8 and 12 hours, the cells begin to migrate. So we see this dynamic activity. But what actually happens before, it's something we can look at more into.
And one more question I have. So when you say the cells undergo migration, so you see increased migratory potential. So I was wondering, what is the nature of these cells? Do these keratinocytes undergo rapid differentiation? Did you observe that?
No, not these keratinocytes. They don't differentiate much during this process. No.
But then, does the differentiation potential get affected later?
Later, yeah, that's something we can look at.
Okay.
It might be interesting, yeah.
Thanks a lot.
Thank you very much.
Awesome, thank you. Ste, go ahead.
Sorry, it just came to mind. Do you know why the movement is towards center? So is it stereotypic towards the center? There is a reason why it's towards the center. Sorry, I completely don't know.
Yeah.
It's epithelial on this. So I mean, actually, I haven't worked with stem cells in vitro myself. So, but I just was wondering why.
Yeah, I often get this question. So in most of our experiments, we see that the cells begin to migrate inwards towards the center of the well. But we do see in some experiments, they migrate the other way. So, but they always begin to migrate highly coordinated and collectively. So that's like the system. So, and what determines if they migrate inwards or outwards, we don't know. We think that the most important thing is that they are organizing themselves in a highly coordinated manner and then decide to move one way or the other.
Okay, so the image, just to follow up to make sure I understand, because I was wondering like what happens to them when they all, do they just pile up in the center and start becoming 3D or something? So those that you showed us, that's not what always happens. There's variability in the the emergent patterns. It's not always the same pattern.
Yes, and when the cells have actually reached the middle part, of course we have increased cell density, and the cells slow down a little bit, but then they actually turn, so they move outwards again. So it's kind of dynamic process. Yeah.
Okay. Yeah, that was my follow-up to Steve's question. Yeah, Denor, do you want to go again, please? Go ahead.
Emma, I have one more question here. Is it possible that cells where you have treated them with actin or myosin inhibitors, can they undergo quiescence?
That's
do you think is that the primary force for it to enter into dormancy?
I think they can probably enter quiescence, but if we block these actinomycin, so the contractility, they probably should. They are probably affected somehow. I can't really figure out how that would.
Okay.
How the results would be from that is interesting. Thank you.
Thanks.
Very cool. Natalie Hussein saying, very interesting talk. Have you tried putting them on soft substrates? And then is there any effect of coating the well with different ECMs?
We have only, to answer the first question, the last part of the question, we have only used collagen coating, so not different ECM adherence proteins. We have tried different stiffness of the gels in our traction force microscopy experiments. We see how this affects the cell sheets velocity, so how fast the cells migrate. That we have done, yes. So.
Okay, great. Thank you so much. Emma, I would love to talk with you more. I do have a few more questions, but I'm actually super late to a faculty meeting right now. So if anybody wants to chime in with last minute questions, please do so now. Otherwise, I'm gonna start the sign-off process.
Yeah, no, it's we maybe can talk more on a later time.
Awesome, that'd be great. All right, so I'll sign off on YouTube. Everyone watching there, see you next week.